Welcome to Generative AI Futures. Today we're doing a deep dive into a really significant piece of research. It's called Tahoe-x1, scaling perturbation-trained single-cell foundation models to 3 billion parameters. We're going to unpack, the nuts and bolts of this model, the math, and why it's potentially such a big deal for things like drug discovery and cancer research.
Yeah, this paper really hits on a core challenge in cell biology, especially for drug development. I mean, if you work with clinical samples, you know the reality. You often don't have much data. Patient samples are hard to get. The assays cost a fortune. And your study groups, your cohorts, they're usually pretty small.
Right. And traditional deep learning models, they just don't perform well when you've only got a little bit of data to train on. They kind of stall out.
Exactly. But that's where foundation models, FMs, come in. They're built differently. They leverage absolutely massive amounts of pre-training data first, so they learn this sort of fundamental grammar of how cells work, and that gives them this amazing ability for few-shot learning. They can generalize from all that pre-training to a new situation, like a specific patient's tumor, even with limited data. That's the edge.
And Tahoe-x1, or TX1 as they call it, is exactly that kind of model. But it's not just general purpose, right? It's specifically aimed at cancer therapeutics. The biggest version is huge, 3 billion parameters.
Yeah.
But you're saying the size isn't the whole story. It's the data it was trained on.
The data is foundational, pun intended, maybe. It's built on the Tahoe-x100 American compendium that's over 100 million single-cell transcriptomes. High-quality data from 50 different cancer cell lines.
Okay, 100 million is a lot.
Mm-hmm.
But lots of single-cell data sets are big now. What makes Tahoe-x100 Mammoth special?
It's the perturbation. This data set is explicitly perturbative. They didn't just observe the cells. They systematically treated these 50 cell lines with over 1,100 different small molecule drugs. So the model is learning directly from how cells respond to being pushed to interventions.
So it's not just seeing correlations, like gene A tends to be high when gene B is low. It's learning causality. If you hit this cell type with this specific drug, this is the likely cascade of gene expression changes. That's what you need for drug discovery, right?
Precisely. You're moving towards understanding the mechanism, which is what you need if you want to actually translate these findings into the clinic. It's about prediction, about cause and effect.
Okay, let's dig into the tech. How does TX1 actually work? It's a transformer, like many large AI models. But how do you feed something as complex as a cell's gene expression profile into a transformer? They need sequences, don't they?
They do. So they represent each cell as a sequence of gene tokens. Think of each token as representing a specific gene. They limit this sequence to a fixed length, up to 2,048 genes in the later training stage. And they include special tokens. There's a Clel's token. Think of that as representing the overall state of the whole cell.
And the really interesting one, the drug token. How does that work? How do you encode a chemical?
Right. That's critical for learning drug responses. They use a separate encoder called fshmenk specifically for the chemical structures. It starts with these high-dimensional numerical representations of the molecules, Morgan fingerprints for the drugs in the dataset. There are about 377 drugs they had fingerprints for initially, and this chemical encoder gets trained alongside the main model. So the model learns to integrate the gene identity, its expression level, and the chemical context all together.
Okay, and the expression levels themselves, gene counts could be all over the place. How did they handle that? Did they just normalize them?
They discretized them. They grouped the raw counts into a set number of bins, B bins in total. But crucially, they used a method, similar to what SCGPT did, that respects the inherent order of those counts. It wasn't just like putting them into random categories.
Why does that matter? Why not just classify expression as, say, off, low, medium, high?
Because expression levels aren't arbitrary categories. 10 reads is closer to 12 reads than it is to 1,000 reads. There's an order, a magnitude. Using a regression objective, specifically mean squared error, or MSE, instead of a classification objective like cross-entropy, forces the model to understand this numerical relationship between the bins. It learns that bin 5 is closer to bin 6 than bin 20. It preserves that quantitative information.
That makes sense. A subtle detail, but it sounds like it could have a big impact on learning the biology accurately.
It does. And speaking of learning, let's talk about the training objective itself. This is where TX1 gets really clever with its dual decoder system.
Dual decoders. Why two? Isn't the goal just to predict the masked gene expression values?
Yes, but they use two different ways to predict those values. And it serves a specific purpose. It's about forcing the model to learn a good global representation of the cell. The first decoder, the gene-aware one, works as you expect. It predicts the expression of a masked gene based on that gene's own token embedding. It focuses on local accuracy.
Okay, pretty standard masked language model kind of idea, but for genes.
Right. But the second decoder, the cellular one, is different. It also has to predict the expression of the masked genes, but it can only use the information packed into that single CL's token embedding. Remember, the TL's token is meant to summarize the entire cell state. This forces that embedding to become incredibly information-rich, capturing the global context needed to reconstruct individual gene expressions.
I see. So one decoder focuses on local gene detail, the other forces the global cell summary, CL's token, to be really meaningful and predictive. That's quite elegant.
It really is. And the overall loss function the model tries to minimize is just a simple average of the prediction errors, the MSE, from both decoders, calculated only on the genes that were originally masked out.
Okay, so that's the architecture and the objective. Now, scaling this up, 3 billion parameters, training on, what was it, 266 million cell profiles in total. That includes Tahoe-100mm plus some other datasets.
Yeah, they pooled Tahoe-100mm with ARC space count and data from CZ-cell-X gene to get that massive scale.
You can't just run that on your laptop. How do they manage the sheer scale and cost?
Yeah.
The paper emphasizes efficiency gains.
Absolutely. This is a key part of the story. They report getting 3 to 30 times better compute efficiency compared to earlier models like SE600M or SCGPT. Essentially, they brought state-of-the-art techniques from training huge language models over to computational biology, very effectively, I might add.
So what were the main tricks, the efficiency hacks?
Well, some are standard practice now for large models, but essential nonetheless. Things like mixed precision training. Using BF16 numbers instead of full 32-bit floats saves a ton of memory and speeds things up, usually without hurting accuracy much. And massive parallelism. For the 3B parameter model, they used fully sharded data parallel, FSDP. That splits the model and its training state across all 128 GPUs they used. Basically, no single GPU holds the whole model.
So clever ways to distribute the load and reduce memory footprint. Standard but necessary.
Exactly. They also used really I/O-efficient data loading methods, things like streaming datasets so the GPUs weren't sitting idle waiting for data. But the real game changer, the architectural insight that unlocked a lot of speed, was about the attention mechanism.
Attention. I remember you mentioned earlier models like SCGPT use custom attention masks. What did it mean?
It meant they put constraints on which gene tokens could attend to which other tokens in the sequence. They thought this was necessary to model biological interactions correctly, but it makes the computation complex and slow. The TX1 team had a realization. Because they were already masking the expression values in the input tokens, maybe they didn't need the complicated attention masking too.
So they just got rid of the custom attention masks.
They did. They switched to standard, dense attention where every token can potentially attend to every other token. And this was huge because it meant they could suddenly use highly optimized off-the-shelf attention implementations like slash attention for the entire sequence calculation. That provides a massive speed up in training throughput.
Wow. So simplifying the architecture by removing a constraint actually enabled tapping into much faster standard hardware optimizations. Classic engineering win. And it seems like this scaling effort paid off. The paper showed performance kept improving with model size.
Absolutely. They showed really clear monotonic scaling trends. As the models got bigger, from 70 million parameters up to the full 3 billion, performance on key biological benchmarks just kept going up. Benchmarks like predicting pathway membership or cancer cell dependencies, it really validated the whole approach. Scaling these models works for biology too, if you do it efficiently.
Which brings us to the crucial part.
Yeah.
Does it actually work for real biological problems? What's the translational impact for, say, a scientist trying to find a cancer drug?
That's the ultimate goal, right? Context generalization. You need the model to make reliable predictions about drugs or genes in situations it wasn't explicitly trained on, like predicting response in a specific patient tumor type.
Let's look at the benchmarks they used. First up was predicting cancer dependencies using depth-map data. They fine-tuned TX1 to predict something called series scores.
Right. Series scores measure gene essentiality, basically. How much a cancer cell relies on a particular gene to survive. A very negative score means knocking out that gene is likely lethal to the cell. And TX1 performed really well, not just at predicting genes that are essential in almost all cancer cells, which is easier, but also at the much trickier task of predicting context-specific essentiality.
You mean like A gene that's only critical for survival if the cell has a specific mutation, say in the KRAS gene. The model picked up on those specific genetic contexts.
Exactly. It learned those subtle context-dependent vulnerabilities. That's highly relevant for personalized oncology. Then, in another benchmark, they tested if the model had learned fundamental biological organization without being explicitly told about pathways. They used the gene embeddings learned by TX1 to predict whether a gene belongs to one of the 50 well-known hallmark cancer pathways from MSigDB, things like avoiding cell death or enabling angiogenesis.
And how did it do there? They used AUPRC as the metric, right?
Yeah.
Area under the precision recall curve.
Yes, AUPRC is good for these kinds of tasks where membership is often sparse. And TX1 achieved the best AUPRC compared to other methods. This suggests the model genuinely internalized biologically meaningful pathway structures just by learning from the perturbation response data. It built its own internal map of cancer biology.
Okay, so it learns dependencies, it learns pathway structure. What about predicting actual drug effects? That seems like the key application.
For that, they combined TX1 embeddings with another model from the Arc Institute, the state transition, or ST model. This combo, TX1 plus ST, was used to predict how gene expression would change after a perturbation. And it worked remarkably well, even in really tough scenarios. It could predict responses in few-shot settings, where you only have a tiny bit of data for a new context. Even more impressively, it worked in zero-shot settings, for example, predicting how blood cells from donors the model had never seen before would respond to treatment with cytokines. That's a really hard generalization challenge.
And they had a metric for this, perturbation separability.
Yeah, that metric basically measures how well the model's embeddings can distinguish between cells that received a treatment versus control cells. TX1 embeddings were significantly better at the separation compared to just using standard methods like highly variable genes.
So what does this enable in practice? What workflows open up?
Two really crucial ones for oncology translation. First, what they call screen-to-patient transfer. You use the model, trained on rich cell line data, to predict which drugs might work in specific patient tumor contexts where you have very limited experimental data. It helps prioritize. Second, context expansion. You might study a drug deeply in a few sentinel cell lines, and then use the model to extrapolate and predict how that drug would likely affect a much wider range of related cell types or genetic backgrounds. It's like having a virtual lab.
Stepping back then, the big picture seems to be that scale matters, but only when combined with the right kind of data. This large-scale perturbation data was key. Just having observational data wouldn't be enough for these causal predictive tasks.
That really seems to be the core message. The causal nature of the perturbation data is what gives the model its translational power. But we should also be clear about the limitations, which the authors acknowledge.
Right. What are the main ones?
Well, the Tahoe-100M dataset, while amazing, is currently based entirely on in vitro cancer cell lines grown in dishes. That's not the same complexity as a real tumor growing in a patient, with its microenvironment, immune cells, stromal cells.
So translating these predictions perfectly to in vivo patient settings is still a major hurdle.
It is. And the model architecture itself, while powerful for gene expression and chemical context, doesn't yet incorporate other important biological data types. Things like protein levels, epigenetic states, or spatial information about where cells are located within a tissue. Integrating those multi-omics data is definitely a big next step for the field.
Makes sense. It's a powerful tool, but still a model focused on specific data types and contexts.
Exactly. But even with those caveats, TX1 is a major step forward. It clearly demonstrates that if you scale foundation models using rich, causally informative data, you get real, tangible improvements in predicting complex biological responses relevant to oncology. They're building increasingly sophisticated virtual cell models.
So the really profound implication here is this potential to bridge the gap, right? Between the simplified world of lab experiments where we can generate tons of data and the messy, complex, data-scarce reality of individual patient tumors. Given how well TX1 did at predicting effects in unseen contexts, it really makes you wonder. So here's the final thought for you, the listener, to explore. How soon might we see these kinds of deep learning models become the standard way we do preclinical drug evaluation and target validation? Could they largely replace the slower, more expensive, traditional screening methods we use today?